everybody, my name is Daniella Olshansky, and in this video I'll be presenting section 26.11, How do enzymes work citrate synthase, using only the material from our textbook. This section explains the general principles of enzyme catalysis, and then using one major enzyme example, citrate synthase, to show exactly how enzymes position substrates, stabilize transition states, carry out acid-base chemistry, and control reactivity inside the active site. I'm going to go through each page of the section that we were provided, explain all the diagrams and mechanisms carefully, and then end with the practice problem that I will solve step by step. So, starting off, how enzymes work. So, this section starts by reminding us that enzymes increase reaction rates by lowering the activation energy, and not by actually changing the equilibrium. They do this through several coordinated strategies. The first strategy is bringing reactants together. In a normal solution, molecules move around randomly and may collide in random orientations. But an enzyme's active site actually brings the substrate molecules into close physical contact, making productive collisions far more likely. The second strategy is holding molecules in the correct orientation. Many organic reactions, especially ones including carbonyls, require the attacking nucleophile to approach from a specific direction. Citrate synthase does this by binding oxaloacetate in a very specific pose before acetyl CoA binds. The third strategy is providing the correct acidic and basic residues. The book explains that inside citrate synthase, a histidine and aspartate residue are positioned so perfectly that they can actually transfer protons at exactly the right time. The fourth strategy is transition state stabilization, and this means that the active site is shaped so that the higher energy transition state is actually more stable inside the enzyme environment, which lowers the activation barrier and speeds up the reaction. The page then introduces obviously citrate synthase itself, it being a global protein of 433 amino acids, and it has a deep cleft where the first substrate, oxaloacetate, will bind. After oxaloacetate binds, the enzyme undergoes a conformational change that closes the cleft and opens a new one for acetyl CoA. The text emphasizes that oxaloacetate will actually bind first, and that order is essential. If acetyl CoA were allowed to bind first, it could actually react incorrectly or even at the wrong time. So this is one of the many examples in biochemistry where the sequence of binding controls the chemistry. So the main message here is that enzymes are not passive. They actively control geometry, proton transfers, and timing in order to lower activation energy. So, a little bit more about this. I just wanted to show basically kind of explaining what this figure is on our page. This slide shows basically the explanation of how citrate synthase actually will work. So the most important concept on this page is actually induced fit. So when oxaloacetate will bind inside the cleft, the enzyme actually changes shape. And this shape changes closes the first cleft and will open a second one that is perfectly shaped for acetyl CoA. So this ensures that acetyl CoA can only enter after oxaloacetate is positioned. It prevents the wrong reactant from entering first, and it also lines up the catalytic residues at exactly the right distance from the substrates. So this is actually a great example of how enzyme catalysis is not only about chemical reactions, but it's about structural control. Enzymes control when a reaction happens, where it happens, and whether it can even happen at all. So this idea of induced fit is a key part of understanding catalysis at the molecular level. So as you can see here, our oxaloacetate and our acetyl CoA will use citrate and synthase, right? Using our synthase, as we were describing, to make citrate. So, continuing on, we are now going to be basically explaining here some structures and functions of some very common coenzymes that we see. That's a lot in our body functions that we see in a lot of our biology classes, such as ATP, coenzyme A. These are really, really important enzymes that are really very important parts of our body. So this is basically going to include a table of important enzymes, but before I walk through these, I just want to add a little bit more detail on what enzymes actually are, but because understanding this helps explain why coenzymes are actually even needed in the first place. So enzymes are biological catalysts, and almost all of them are proteins composed of long chains of amino acids folded into highly specific three-dimensional shapes. This precise folding will create the active site, which is a pocket or groove where substrates bind and where the chemical transformation will take place. One important thing to understand is that while amino acids are incredibly versatile, they still have a lot of limitations. So they can either donate or accept proteins, hold substrates in place, and stabilize transition states. But there are certain chemical reactions, especially ones that require unusual functional groups, metal ions, radical chemistry, or carbanion stabilization, that amino acids alone can't really perform efficiently. And that's actually where coenzymes come in. Coenzymes are small organic molecules often derived from vitamins that sit in the active site along the enzyme and provide specialized chemical abilities that the amino acid side chains do not have. So together, the enzyme and the coenzyme act like a team, right? So the enzymes are going to provide our structure, the orientation, that protein environment, but that coenzyme is going to provide that unique chemistry that's actually needed for that reaction. So even though citrate synthase itself does not require an enzyme, it only uses amino acid side chains for catalysis. And the textbook includes this table to show that many enzymes do require these additional helper molecules. So I'm going to go through now a couple of these coenzymes listed on the page, starting with our tetrahydrofluorate. It's going to be here. It transfers one carbon fragment in metabolic pathways, especially in nucleotide synthesis. We have PLP, which stands for pyridoxal phosphate. Our PLP is right here. As you can see, it's an amino acid metabolism, right? It's actually essential in all of our almost amino acid chemistries, including transamination, decarboxylation, and racemization. We have lipoic acid, which is going to be participating in a lot of our redox reactions and transferring acyl groups. And it plays a key role in pyruvate dehydrogenase complex. And we actually used lipoic acid in one of our past experiments that we actually did this semester. And as well as thiamine diphosphate, which is TPP, extremely important for decarboxylating alpha keto acids, a reaction normal amino acids actually are not allowed to do alone. And biotin, a classic carbon dioxide carrier used in carboxylation reactions like the conversion of pyruvate to oxaloacetate. So the point of this page is just to broaden the definition of what enzymes are, right? An enzyme is not just the protein, it's the protein plus any coenzymes or cofactors as it requires to function. And then also we have a couple here, right? ATP, we know ATP is one of our main ways that we have energy. Coenzyme A, another way that helps us actually to produce functions in our protein environments. Same thing with NAD+, right, in our oxidation reductions, as well as flavin adenine dinucleotides as well, used in a lot of our oxidation reduction experiments that we actually do. So and continuing on, so we have here a picture of our citrate synthase structure. So this is actually going to be one of the more visually informative pages here. And so starting with section A, as you can see, this is a space filling model. And this basically shows the enzyme as a solid 3D object, right? It highlights how packed the protein structure is and emphasizes how the active site cleft is carved out. And this dense packing, as you can see, right, prevents water and other molecules from interfering with that reaction. And then in B, we have the ribbon model, right? So this diagram is going to focus on our secondary structure, mostly alpha helices, and it also illustrates that citrate synthase is a dimer, which means that two identical protein chains will associate tightly to form the actually full enzyme. And dimerization also often will increase stability and allow the subunits to assist each other during catalysis. And our final one in part C, as you can see, it is the active site, and it's actually giving us a close-up of what this looks like, right? So this is going to be probably the most important part on this page. It's oxaloacetate and acetyl-CoA mimic are bound inside that active site, right? And you can clearly see here histidine 274 and aspartate 375, these catalytic residues are going to carry out that acid-base chemistry. And histidine donates or accepts protons at different steps. And this aspartate will remove that alpha proton from acetyl-CoA to generate that enol. So the way these residues are positioned, not just in the right location, but at the right distance and orientation is what allows this reaction to proceed so efficiently. And this structural page will bridge the gap between the introduction and the mechanism. So now that we've seen where the substrates bind and where the catalytic residues are, we can now understand the reaction mechanism pretty clearly. So this is going to be our full mechanism. So we're going to show the complete citrate synthase mechanism. So starting off, step 1, we have enol formation, right? So aspartate acts as this base, and it'll remove the acidic alpha proton from acetyl-CoA, as you can see. At the same time, a histidine residue, right, will donate that proton to the carbonyl oxygen, right? And you can see that. We can see that it's going to generate our enol, right? So these two simultaneous events will generate the enol, which is very, very necessary for this nucleophilic attack to happen. And this is basically going to be our first step, right? So as you can see, we formed our enol. We had we need to have this to occur for our nucleophilic attack. And basically overall, right, you can see we have our NH group, right, on that side chain of histidine acting as the acid. And then what do we have? We have that aspartic acid acting as our base. So step 2 is now going to be our aldol-like addition, right? So the enol attacks the carbonyl carbon of oxaloacetate, forming the new carbon-carbon bond, as you can see, right? So that second histidine is going to donate a proton to stabilize that tetrahedral intermediate, forming citral CoA. And this step will mirror an aldol reaction, but is controlled entirely by the enzyme's environment, right? So here we're going to form, right, our S-citral CoA, showing that it has that stereochemistry, right? Showing that positions are really, really important here for actually to form that citrate, as we said. And our final thing here is our hydrolysis, right? Adding water. So water will perform now our nucleophilic attack on the thioester of that citral CoA, as you can see, right? And the cleavage of this thioester will produce citrate and obviously release coenzyme A, which we just talked a little bit before of some really common examples of coenzymes that we produce. And this hydrolysis step is energetically favorable and will drive the entire reaction forward. So this mechanism demonstrates all fundamental principles of enzymatic catalysis, precise orientation, acid-base chemistry, induced fit, and transition state stabilization. So finishing off, we have a practice problem. So obviously using figure 26.11.2 from our textbook, we want to identify which amino acid will act as the base to remove that alpha proton from acetyl CoA and explain why that proton is acidic enough to even be removed. So let's do this. So looking at this, I'm going to basically show you guys. So this amino acid, okay, that acts as the base, as we can see, is going to be our aspartate 375, which is what we looked at a couple slides ago, right? And we said that that side chain is going to be our negatively charged carboxylate, okay? So this residue will be perfectly positioned to remove the alpha proton from acetyl CoA during step 1 of the mechanism, right? So we see that the residue is perfectly positioned to remove the alpha proton from that acetyl CoA, right? We're removing that alpha proton, and it's going to form acetyl CoA. So this is the first part. This is basically going to be what is acting as the base, aspartate 375. Now, the alpha proton is going to be acidic, right? So it's going to be acidic because removing it will form our enol, right? And it's going to be resonance stabilized. The negative charge, right, as we know, can be delocalized onto our carbonyl oxygen, as we see here, right? Additionally, the enzyme's active site will stabilize the developing negative charge through hydrogen bonding and orientation. And so that's basically going to be, our correct answer is aspartate removes the alpha proton because the resulting enol is resonance stabilized and further stabilized by the enzyme environment. And this is really it. Thank you guys so much for watching my presentation on section 26.11, how do enzymes work. Everything in my video was taken directly from the textbook pages provided, and I hope this helped make the mechanism and concept clear. I hope you have a great day. Thank you so much.